All right, so I'll do a very brief introduction of tonight's speaker. First, I want to welcome all of you. This is the first of a series of lectures in the Molecular Medicine Evening Lecture program. And this is the second season that we've done this. And this is a series that's sponsored by the Molecular Medicine program at the University of Washington. This is an Howard Hughes Institute-funded program. And the goal of this program is to bring some of the excitement at the interface between biology and medicine both to you as an audience and to incorporate some of that into PhD biomedical research training on this campus. So this is a program that's been in existence now for several years. I see several of the participants in that program in the audience, and it's an opportunity, in addition to getting a PhD in one of a dozen different disciplines at this institution, to have contact with individuals that are trying to bring basic science to the clinic and also take clues from the clinic to improve our understanding of basic human biology and human disease processes. So the lecture series is an attempt to try to pick individuals who have participated in this course, such as this particular series of courses, such as Jay, to talk about what they're doing in research and what they see as some of the most exciting aspects of the research they work on and how it might influence both patient care and our understanding of human biology. So our speaker tonight, Jay Heinecke, I've known for quite a while, and I told him I'd not tell any embarrassing stories. But Jay originally did his bachelor's degree at Antioch. He went to Washington University to do his MD after that, and then came here to the University of Washington to basically finish his medical training. He did a medicine residency and internship, and he did an endocrinology residency. And he also did something that was a little bit unusual. He did a research fellowship in biochemistry, and it was there that he got interested in a topic that will play in tonight's talk, which is the role of oxygen in biological systems, and especially some of the potential deleterious aspects of oxygen as they affect proteins in our blood. So this is part of the theme of this evening's discussion. In 2002, actually in 1991, when he finished his training here, Washington University, where he had done his medical training, realized this was a good thing. They promptly hired him back as an assistant professor, and he was there through 2002 when we were lucky enough to recruit him back to this campus as a professor of medicine. And he's been here since 2002, and since 2004 has been the Karasinski Chair in the Department of Medicine in the Division of Endocrinology. So it's a great pleasure to welcome Jay to this series, and I look forward to tonight's talk. Jay.
Well, thank you very much, Ray, for that kind introduction. The real reason he's not telling any stories about me is that he doesn't want me to tell any stories about him. So it's kind of a quid pro quo here. So what I'd like to talk about tonight is HDL, or high-density lipoprotein. And what I hope to convince you about at the end of the evening is that maybe we know far less than we think we do about this fascinating particle. So what I'd like to do is to start off with the key players in this drama. These are highly detailed molecular models that my colleague Jack Oram constructed, delineating the structure of these particles in the blood. So on the left side, we have low-density lipoprotein, or LDL cholesterol. This is the bad form of cholesterol. And on the right side, we have the good player, HDL cholesterol, or high-density lipoprotein. So what is the evidence that these proteins and lipids are important in biology, and why do we call these the good and bad forms of cholesterol? Well, the original data substantiating these ideas was based on isolating these particles from the blood of human beings and looking at them in people who did and didn't have heart disease. And what rapidly became apparent was that when people had high levels of LDL cholesterol, they had a greatly increased risk of coronary artery disease. And this was first demonstrated over 30 years ago in epidemiological studies, but has been confirmed over and over and over again in many different ways. It was subsequently discovered that there was also a good form of cholesterol in blood, or high-density lipoprotein. And when levels of this form of lipids and proteins are elevated, your risk for coronary artery disease goes down. So probably most people in the room are aware of the idea that LDL is bad for you, but I'll point out that in men under the age of 60, a low level of HDL cholesterol is a more common risk factor for heart disease than a high level of LDL cholesterol. So I guess one of the questions that always comes up is, why do we have these lipoproteins if they're so potentially dangerous? And like all things in biology, and I was discussing this with the students earlier, there's really one driving force for this, and this is sex. If you don't reproduce and pass on your genes, you're a biological dead end. You really don't matter as far as the organism is concerned. And there are really two key things to being able to have sex, to reproduce. You have to have adequate nutrition or food intake, and you have to be able to resist disease. And through most of human history, the major killers of human beings was infectious disease. And so the reason we evolved lipoproteins, I'd like to suggest, is to play a key role in nutrient delivery and disease fighting. So the underlying reason for generating these particles is that fat does not like to be in water. It's what we call hydrophobic. And so you have to deliver these particles in a very special way that I'll go into in just a minute that involves both proteins and lipids. So there are two key components of lipids or fats that are relevant to lipoprotein biology. And these are triglycerides, which we abbreviate TG. These are very nutrient-rich particles that we have to transport from our gut to our adipose tissue. The other key component of these particles is cholesterol. Now, cholesterol is not a nutrient in the traditional sense, but it plays a very key role in maintaining the integrity of membranes around cells. In fact, if you lack cholesterol, your cells are leaky, and this is a lethal phenotype in vivo. Cholesterol also plays many other roles, including the formation of hormones like estrogen and testosterone. So the problem that we confront with lipids is that we have to transport these insoluble fats through the blood in the aqueous or water environment. So the way that your body solves this problem is to make particles that are composed of both protein and lipids. And in this somewhat oversimplified model, the fats, like triglycerides, partition into the core of these spherical particles so that they avoid interacting with water. In contrast, the proteins that help solubilize these particles and also play a very important role in controlling their biological properties reside on the surface of the particles and help solubilize the insoluble fat that resides in the center of the particles. So what are diagrammed here are actually four different classes of lipoproteins. Chylomicrons, which are very large, fat-rich particles that deliver fat from your diet to the adipose tissue via the liver. On the right side of the figure are VLDL and LDL, which also play a very important role in delivering fat to adipose tissue. And then on the bottom left is the HDL particle, the high-density lipoprotein particle. And the reason that these particles are high density is they contain far more protein than lipid, and this makes them denser than the other particles. Okay, so what's the underlying rationale for this physiologically? So this has been the dogma that has driven our concepts of lipoprotein metabolism for the last 30 years. So we basically start with a nutrient source rich in fat, in our culture perhaps a Twinkie, and our goal is to get the calories that are in this nutrient to your adipose tissue. So initially we take up the fats into our intestines, and those fats are absorbed. But we don't transport them directly to adipose tissue. Instead, we send those fats to the liver via these very large particles that contain a lot of triglyceride called chylomicrons. The particles are disassembled in the liver and reassembled into VLDL and LDL that are secreted by the liver, and those particles actually transport the nutrient-rich triglycerides to the adipose tissue. So one of the paradoxes of this is that these particles are very rich in cholesterol, which, as I've told you, is a key player in maintaining the integrity of your membranes so that they don't leak water. But the problem with cholesterol, the yin and the yang of cholesterol, is that it also can promote inflammation in a kind of a white blood cell that exists in your body called a macrophage. So macrophages are white blood cells that play a very important role in fighting infection. And when they acquire too much cholesterol, they become angry and they do bad things. An example of that is an atherosclerotic lesion. So this is what kills most people in the United States. About 40 to 50% of people in the United States die from coronary artery disease. And the underlying problem is accumulation of cholesterol in the arteries that supply blood to your heart. An example of this is shown in this picture right here. This is a atherosclerotic lesion, very lipid-rich, rich in cholesterol, that's ruptured and triggered the formation of a clot in the artery, killing the patient. So this is what causes a myocardial infarction or a heart attack. So this is illustrating kind of our current concept for how macrophages, which are a very key component of the atherosclerotic lesions, these white blood cells, get into the artery wall and accumulate cholesterol. So the initial event is thought to be the bad form of cholesterol, or LDL, getting into the artery wall. And we actually still don't understand exactly how this takes place. Subsequently, again by poorly understood pathways that may involve oxidative stress, the cholesterol triggers the secretion of inflammatory hormones and signaling molecules that lure monocytes, the precursors of macrophages, into the artery wall. So these are inflammatory white blood cells that normally would fight infection, but in the setting of too much cholesterol in the artery wall, they sense that inflammation is going on and they enter that environment. And there they take up too much cholesterol to become cholesterol-laden foam cells that are the key cellular player in atherosclerosis. Okay, so how do LDL and HDL work in this scenario? So the traditional idea is that LDL is very rich in cholesterol and that it binds to scavenger receptors on the cell surface of the macrophage. These are receptors that were originally developed to fight invading pathogens, but because LDL has become modified, it now is recognized by these receptors and delivers cholesterol in an unregulated manner to the cell. And this produces what we call a foam cell. This is a macrophage that's become cholesterol ester loaded. And these are angry macrophages. They're not happy. They produce inflammatory mediators and they do lots of bad things. In contrast, HDL traditionally is thought to remove this excess cholesterol. And so the idea would be that HDL counteracts the bad effects of LDL by removing cholesterol from the macrophages. So the idea is, I think, and this is a very important idea, is that it's not really LDL itself that's necessarily bad for you. It's a modified form of LDL that can bind to these receptors on macrophages that normally recognize invading pathogens. And I think one of the themes of the story that I'd like to tell you tonight is that we think that HDL is also playing a role in these infectious pathways. So the notion is that LDL is modified and that causes disease. So one very attractive hypothesis that's supported by many different lines of evidence is that this modification reaction that occurs to LDL occurs via a pathway we call oxidative stress. And I'll give you an example of how this might work in just a minute. But the notion is initially LDL gets into the artery wall and then somehow mysteriously becomes oxidized. And it's this oxidized form of LDL that actually lures the monocytes into the artery wall to become macrophages. In concert with that, oxidized LDL can also bind to the cell surface of these macrophages and be taken up, producing cholesterol-loaded macrophages. And it's these foam cells that are actually the critically important link in promoting inflammation in the artery wall and lesion formation and plaque rupture. So one pathway that white blood cells like macrophages use to generate reactive intermediates involves an enzyme called myeloperoxidase, or MPO. And I've colored it green here because this is a beautiful green protein. The first time that I purified this, there was an intense green column of the protein bound to the column that I was using for the isolation procedure. And as soon as I saw this protein, I knew I wanted to work on it. It's an incredibly beautiful protein. So normally, the myeloperoxidase resides in granules inside macrophages and other white blood cells. But when the cells become activated, for example, by sensing a bacterial or fungal pathogen in their environment, this triggers activation of the cells. And two things happen. First, the cells use a membrane-associated enzyme system to generate hydrogen peroxide, a relatively weak oxidant. The cells also secrete myeloperoxidase. And the myeloperoxidase interacts with the hydrogen peroxide to generate an oxidant called hypochlorous acid. And probably this is more familiar to people in the room as bleach. So we use bleach, for example, to sterilize pools or our water supply. And what we're basically doing is recapitulating an enzyme pathway that was developed hundreds of millions of years ago by white blood cells to kill invading pathogens. So your white blood cells make bleach. So the normal role of this pathway is to kill bacteria and fungus. And we've been able to use mice that lack myeloperoxidase to demonstrate this. But what we proposed when I originally moved to Washington University was that bleach could also be a way to damage host tissue at signs of chronic or acute inflammation. So we originally became interested in the idea then that bleach generation by myeloperoxidase might be a mechanism for the macrophages to damage LDL and promote foam cell formation. So to first test this idea, we did a very simple experiment. What we're looking at here is human atherosclerotic lesions that have been stained with antibodies that recognize specific things. And the staining is demonstrated by the intense red color that you see. So on the left panel, we're looking at a region of the lesion that's very rich in macrophages. And on the right side of the panel, we're actually looking at something we call the necrotic core that doesn't have many cells in it, but is very rich in cholesterol. In the upper panel, we're using an antibody that recognizes oxidized LDL. So notice that it stains the macrophages, and it has a very distinct immunostaining pattern in the advanced acellular lesions on the right side as well. In the bottom panel, we've used an antibody that recognizes myeloperoxidase to stain the lesions, and these are serial sections in the same lesions. And what you can see is a very strikingly similar pattern of staining. Most of the MPO is associated with macrophages in the macrophage-rich part of the lesion, and most of it is around these cholesterol clusters, or crystals as we call them, in the acellular necrotic core. So based on these and other observations, we proposed that myeloperoxidase might be one mechanism for oxidizing LDL in the human artery wall. So this is demonstrating the staining for oxidized LDL in the top and for myeloperoxidase in the bottom. Okay, so the notion would be then that MPO is has a critical role in modifying LDL to a form that can bind to scavenger receptors and promote foam cell formation. And all of these events have been mimicked in the test tube using LDL and myeloperoxidase in macrophages. So when I moved from Washington University to the University of Washington, we became interested in the idea that HDL, or the good form of cholesterol, might also be targeted for oxidation. And this is surprisingly something that no one had ever really considered before. Almost all the previous work on oxidative stress and damage of lipoproteins had centered on LDL. So one of the ways that we investigated this question was to measure a very specific product of bleach. So one of the great things about studying bleach is that it's a very unusual oxidant. It contains a reactive moiety called chlorine. Chlorine can react with molecules to form chlorinated biomolecules. And so what we did was to develop assays, very specific and sensitive mass spectrometric assays that could detect chlorotyrosine in human biological material. And then we went on to use this technique to look at HDL isolated from humans. So the notion would be this chlorinated tyrosine is a very specific molecular fingerprint that helps us identify whether or not this chemistry is taking place in the human artery wall. So this is just illustrating the chemistry again. Myeloperoxidase reacts with peroxide to form bleach, and bleach chlorinates an amino acid called tyrosine to form 3-chlorotyrosine. And then, in turn, we suspected that the bleach might react with the major HDL protein, which is called ApoA, to form chlorinated tyrosine residues. So the initial experiment was actually a very simple one. We isolated HDL from the blood of either people with established coronary artery disease, abbreviated CAD, or age and sex matched control subjects, and then we quantified the levels of 3-chlorotyrosine in the HDL using isotope dilution gas chromatography mass spectrometry. And we really got a rather striking result. When we looked at the patients with established heart disease, every single subject had detectable chlorotyrosine in their HDL. In striking contrast, except for one individual, chlorotyrosine was undetectable in the HDL of the subjects who were disease-free. So based on these and other studies, we proposed that HDL was also a target for oxidative damage in humans with coronary artery disease. So one of the ideas for what HDL is doing is, as I said earlier, is that it's removing cholesterol from macrophages. And so it was an attractive hypothesis to us that this kind of chlorinated damage might block the ability of HDL to remove cholesterol from cells. And so we went on to investigate this question with our colleague Jack Oram, who provided those highly detailed molecular models of good and bad cholesterol that I showed you earlier. And we focused on a specific protein in macrophages called ABCA1. So this is a simplified view of how HDL removes cholesterol from macrophages. So the pathway starts with the release of lipid-free ApoA1, the major HDL protein from HDL. So most ApoA1 in the circulation is associated with HDL, this lipid protein complex. But it turns out that that complex will not remove cholesterol from cells by this pathway. Instead, the ApoA1 has to come off of the HDL particle. In turn, this, what we call lipid-free ApoA1, interacts with a protein that's in the membrane of the cells to promote the efflux of cholesterol and other lipids in the membranes from the cells. And so what happens is these cells actually generate little lipoprotein-like particles at their cell surface that are taking away the cholesterol together with other lipids from the cell surface. And this in turn depletes the macrophages of cholesterol. We know this pathway is important in humans because when you lack this ABC protein, you suffer from Tangier's disease. And in this disorder, you accumulate cholesterol-laden macrophages in many different tissues in your body. And we can also mimic this disease in mice by knocking out the ABCA1 pathway. And when we do this just in macrophages, we dramatically increase atherosclerosis. So this is illustrating in a little bit more detail how we think this is working. So as I emphasized earlier, it's a lipid-free form of ApoA1 that has to interact with ABCA1. And this is actually a representation of a more detailed model of that structure based on X-ray crystallography. And the key feature here for you to observe is that it is composed of a 4-helical bundle. So if you look carefully at the left side of the figure, there are 4 bundles that run next to each other in an anti-parallel conformation. This is a key structural component of the lipid-free HDL. In contrast, when ApoA1 is bound to HDL, it has a very different conformation. Now it looks like a horseshoe-shaped molecule, and one could just envision this molecule going around a spherical particle and accompanying it. So what Ages et al. Proposed in a paper that was published several years ago was that the key step in lipid-free ApoA1 removing cholesterol from cells was a reorganization of the lipid-free ApoA1 from this 4-helical bundle structure to this horseshoe-shaped structure. And they proposed that remodeling of the particles in the hinge domains was critical for this pathway. So what we were able to demonstrate in a whole series of studies that I won't have time to show you tonight is that specific amino acid residues in these hinge domains of ApoA1 were specifically targeted for oxidative damage by myeloperoxidase, and that this blocked the ability of the chlorinated ApoA1 to remove cholesterol from the cells. And so the idea is that by targeting very, very specific amino acid residues in this protein, we can disrupt its biological function. Okay, so just to summarize the story so far, What we would argue then is that myeloperoxidase, which is actually secreted by the macrophages themselves, can oxidize LDL, and this causes it to bind to the scavenger receptors and deliver cholesterol. And in parallel, it blocks the ability of HDL to remove cholesterol from the cells. And this kind of double whammy plays a key role in promoting macrophage foam cell formation in the artery wall. Okay, so the traditional notion then is that HDL acts by removing cholesterol from cells. But more recently, a completely different idea has developed, and this is that HDL may also be a very important regulator of inflammation. And there is evidence in both humans and mice, using, again, knockouts of specific proteins, that you can actually make forms of HDL that promote atherosclerosis or heart disease. And we call this dysfunctional HDL. So this is when HDL goes bad. And no one really knows what the molecular basis for this is, but a number of different pathways have been proposed, possibly involving oxidation. And I've already given you examples of how that might work around macrophages in the artery wall by damaging the ability of ApoA1 to remove cholesterol from cells. And another attractive hypothesis is that we're changing the protein content of HDL, shifting the balance between pro and anti-inflammatory effects. So a number of lines of evidence strongly support this idea in mouse models of atherosclerosis. For example, if you knock out ApoA1, the major HDL protein in mice, and you make these animals have a high level of LDL cholesterol, they develop a systemic inflammatory response, and male mice actually go on to die from this disorder. And similar phenotypes, as we call them, or similar manifestations have been described in macrophages that have other defects in excreting cholesterol. And I think from a clinical perspective, very excitingly, in animal models of arterial inflammation, you can infuse HDL and actually block the white blood cell inflammatory response, suggesting that this might also have therapeutic implications in humans. So we became very interested in this area, and we thought that one potential way of trying to understand what this dysfunctional HDL was and how HDL might have anti-inflammatory effects was to look at the protein content of HDL. So proteins in HDL play a very important role in solubilizing the lipids, but they also have very powerful biological effects. And I've already mentioned one, which is the ability of lipid-free ApoA1 to remove cholesterol from cells. But there are many, many other potential biological effects. And so in order to try and understand this better at the molecular level, we again took advantage of mass spectrometry, in this case to analyze the protein composition of HDL in a very comprehensive and unbiased manner. So this is showing you the results of our initial studies of HDL, and this was a big surprise to us. We were able to show that HDL actually contains at least 48 different proteins, and this was about 15 more proteins than had previously been known to reside in HDL. So HDL is not just ApoA1. It's ApoA1, the major structural protein, but a whole array of other protein components. Not all of these proteins are on the same particle. That's not what I'm arguing. In fact, what we really think is that this is a family of particles with very distinct protein composition. When we analyzed what these proteins were, as we anticipated, we had found every protein that had previously been implicated in lipid metabolism except for one, a protein that's present at very low concentrations in HDL. So this was very nice because it was a positive control. It meant that we could find what was already known to be present in the HDL. The first big surprise was there are more acute phase response proteins in HDL than proteins involved in lipid or fat metabolism. The acute phase response is something that happens in animals and humans when they have an infection or a fever, and it dramatically changes the protein composition of your blood, we think to fight off invading pathogens. The fact that so many proteins in HDL are regulated in the acute phase response strongly implies that it's involved in the inflammatory pathway. We also found two protein families that had not previously known to reside in HDL. a series of proteins that are called proteinase inhibitors. Proteases are proteins that degrade other proteins. This is very important in host defense mechanisms, but it's also thought to play a role in triggering plaque rupture and clot formation in atherosclerosis. And so this again links HDL both to inflammation and potentially to atherosclerosis. And then another set of proteins that regulate complement. And complement is a family of proteins that reside in your blood that constitute the first line of defense against invading pathogens. So we think that collectively these observations suggest that HDL plays a very important role, not just in lipid metabolism, but in inflammation and host defense mechanisms against invading pathogens. There's already abundant evidence in the scientific literature that supports this hypothesis that's been largely ignored by us lipoprotein biologists. I think a particularly interesting example is something that was originally described as a trypanosome lytic factor. So a trypanosome is a parasite that resides in certain regions of Africa, and it causes a very specific disease in humans. And what was discovered was that there was a factor in blood that could kill the trypanosome. And this factor was subsequently shown using biochemical approaches to consist of ApoA1, the major HDL protein, another protein in HDL called haptoglobin-related protein, and another protein called ApoL. And in the test tube, we can reconstitute this killing system just by mixing the purified proteins together. But I think a very key point here is these proteins reside in a very specific complex in HDL. They're not floating around. This is not a random distribution of these proteins. These three proteins exist in a specific macromolecular complex. Well, the relevance of this to human biology was not really clear. This was all test tube biology. But as I've emphasized, a key component of this killing complex is ApoL. And 2 years ago, a case report appeared in the New England Journal of Medicine of a human who was infected with a form of trypanosomes that has only been observed to infect cows before. And when they went back and looked at this person, it turned out they had a mutation in ApoL, and that they had no ApoL in their HDL particles. And so these and other lines of evidence would suggest that, in fact, certain subspecies of HDL can play a key role in host defense mechanisms against invading pathogens. So this is kind of illustrating what we think is a paradigm shift in thinking about HDL. We have the old notion, based on the nutrient delivery hypothesis, that the major function of LDL and VLDL is to deliver these nutrient-rich triglycerides from the liver to the adipose tissue. And in this scenario, HDL is kind of a passive player that just accepts cholesterol from macrophages and takes it back to the liver for excretion. In our model, we have a very different way of thinking about this. We think that HDL is inextricably linked to macrophage activation and host defense mechanisms. And we don't think that this is just kind of a passive process where cholesterol comes off the surface of the cell and adsorbs onto HDL. We think that it's a dialogue between the HDL and the macrophages, and that this in turn plays a very important role in controlling inflammation and perhaps in controlling infection in humans. And there's already strong evidence from many other investigators that proteins can jump from macrophages onto HDL, consistent with this model. And so what we would speculate is that proteins and lipids are being exchanged between HDL and macrophages, and this is playing a key role in regulating how macrophages respond to inflammation and infection. So another interesting component of this hypothesis that the proteins in HDL can be very specialized and have many different functions is that they might also be playing a role in heart disease. So this is coming back to the idea that there could be dysfunctional HDL in humans. And so we thought it would be interesting to look at the protein composition of HDL isolated from humans with established heart disease and control subjects. And again, we used mass spectrometry to do an unbiased analysis of the protein content. So this is a fairly complicated experiment, and I don't have time to go through all the details here, but this is showing you all the proteins that we detect in HDL. And on the bottom is an axis for whether or not the proteins are enriched in one group of subjects. So if a protein has a value of 1 on the bottom axis, it means it's only found in people with heart disease. If it has a -1 value at the top of the graph, it means it's only found in people who are healthy. And if it has a value of 0, it means it's equally abundant in people with and without heart disease. And what you can see is that there are 5 proteins that appear to be enriched in the HDL of people with heart disease. And interestingly, 4 of these 5 proteins are secreted by macrophage foam cells. And so this is a relatively small study. Mass spectrometry is not an easy thing to do in a high throughput form, but we're very intrigued by the idea that altered protein composition in HDL may be a marker and perhaps a mediator of heart disease in humans. So to summarize then, we think that normally HDL is cardioprotective by at least 2 distinct pathways. One involves the removal of cholesterol by ABCA1 and other pathways. This decreases macrophage sterol content and makes them less angry. We also think that proteins in HDL are regulating inflammatory pathways in macrophages. And we have very strong evidence using array analysis of macrophages that this hypothesis is correct. We also like the idea that there could be dysfunctional forms of HDL in humans. And I've outlined some of our work on oxidative damage of ApoA1, and the notion would be there that by damaging specific amino acid residues in ApoA1, we block the ability of A1 to remove cholesterol from macrophages, and this increases their sterol content, their cholesterol content, and makes them angry. We also believe that altered protein, and I should mention perhaps lipid composition of HDL, this is not something we've looked at but we're very interested in, also can play a role in regulating inflammation. And we would speculate that alterations in the protein or lipid content of HDL may also be playing a pathogenic role in atherosclerosis. So where do we need to go with this? What are the big questions? Well, one question that fascinates us, that may seem trivial to many people, is just what is HDL? The way that we traditionally isolate HDL is to put it in a very concentrated salt solution and to spin it at very high centrifugal force for 48 to 72 hours. And it's long been known that this dramatically alters the protein content of HDL. And I've already mentioned this idea that what we think HDL actually is a whole series of specific protein-protein complexes. And so what we speculate happens during this ultracentrifugation step is analogous to having a whole family of Tinkertoy type constructions or specific macromolecular complexes that are essentially randomly dissociated. A third of the proteins are thrown away, and then you just add them back together into some kind of scrambled mixture. So we're very interested in using other biochemical techniques to isolate HDL in ways that don't destroy the particles and to go back and ask about some of these fundamental questions about macrophage biology. We're extremely interested in the idea that HDL is talking to macrophages and regulating inflammatory pathways. And we don't think that this is relevant only to atherosclerosis. Many different diseases involve angry macrophages. Alzheimer's disease is characterized by angry macrophages. Many forms of inflammatory bowel disease are characterized by macrophages. Lung disorders, macrophages. In fact, we've been looking at the protein composition of macrophages, and the proteins that we find secreted by foam cells are also highly abundant in Alzheimer's disease plaques. So we're very interested in the idea that HDL is regulating novel inflammatory pathways in macrophages. And finally, we think that by looking more carefully at the protein composition of HDL, we might be able to come up with a better way of diagnosing people at risk for heart disease. So although everyone gets their LDL and their HDL measured on a routine basis to try and assess cardiovascular risk, it turns out that they're not very strong predictors of risk in individual people. In fact, you can do almost as good by flipping a coin as you can by measuring an HDL and an LDL in an individual. And so we're very intrigued by this idea that by looking more carefully at specific protein components of HDL, we might be able to come up with tests that tell us a lot more about who's at risk for heart disease and whether or not our therapies are working. I'll just mention, for example, that our best therapy for preventing heart disease. Statin therapy, which is one of the biggest successes in medicine today, only prevents a third of heart attacks. And we have no idea how to identify the people that are benefiting, and we also don't know whether or not changing HDL could further improve those results. And so we think that this lipoprotein-protein complex that's long been thought to just be playing a passive role in transporting nutrients actually may have far more fascinating properties, and that there's a long way to go in terms of understanding what's happening with these particles. So with that, I'm going to end my talk, and I'd be happy to take any questions, assuming there are any questions. Let me just thank the people that do the work. So all I do is write the grants and the papers and give the talks, right? So I'd like to specifically mention Bohai Xiao, who's done all of the work on the site-specific modification of ApoA1. Patty Green, Andy Hufnagel, and Tomas Weiser, who've been involved in our proteomics analyses of HDL. And I've really only been able to touch the tip of this iceberg. We have a whole lot of data on this that I wasn't able to share with you tonight that's unpublished. And then Jack Oram, who came up with that detailed molecular structure of the good and bad forms of cholesterol that I showed you at the beginning of the talk, and who does a lot of our macrophage biology, because I'm a chemist at heart. So again, thank you all very much, and if there are any questions.
So hopefully I understood what you were saying, but I'm wondering, are you thinking that there's multiple dysfunctions and individuals might have one or another one, but they're all somehow related to this process?
I think that's a very attractive notion. So we know that people get heart disease for all different kinds of reasons, right? It looks simple. The final thing that you get in an atherosclerotic lesion is a pretty stereotypic response of the artery wall to damage. But we know that in individuals, it's not easy to predict what's happening. And so one very attractive idea is that different things are going on in different people. For example, diabetics have greatly increased risk for heart disease. It's not clear why that is. Simple things like how good their blood glucose control are don't really predict whether or not they're at risk. And so one idea might be that there are differences in the protein composition of the HDL in diabetics compared to non-diabetics. And in fact, we have very strong evidence that that is the case. And so one could imagine many different molecular functions and abnormalities going on. One of the great things about using mass spectrometry to look at the HDL is we can interrogate all of these proteins at one time in one analysis and potentially gain information about many different risk factors in each individual. So we like that idea, and we're pursuing that notion very aggressively in the lab.
Okay. And also, do you think that there may be, or is there any evidence that there might be an association between level of HDL and dysfunction?
Well, see, so this is one of those issues. So our clinical laboratories like simple tests, okay? And so the test for HDL cholesterol and LDL cholesterol are very simple, very profitable, easy to do, anyone can understand it. But what I didn't tell you about was that last year there was actually a clinical trial looking at an HDL elevating agent called torcetrapib. And the trial was stopped early after only 1 year because more people died in the group that got the drug that raised HDL levels. So this is a very fascinating study. Everyone in the study was on statins, so that's our current best therapy for heart disease. Everybody in the study had heart disease, so they were at high risk. What happened was the people who got the HDL elevating drug actually died more from heart disease, died more from cancer. They didn't get more cancer, but if they had cancer, they died more. And very interestingly, there were 8 patients that died from sepsis in the group that got the HDL elevating drug and none in the control group. So many people believe that this reflects the generation of a dysfunctional form of HDL that's actually not preventing inflammation. There are other potential explanations. I want to emphasize that. This drug has other effects. It tends to raise your blood pressure a little bit. It has effects on aldosterone and potassium metabolism, and so there are other hypotheses. We call that off-target toxicities that may explain this. But my colleague Jack Oram actually predicted 10 years ago that this drug would kill people.
Thank you.
So we'll see.
I was curious if the mass spectrometry work you've done with HDL proteins, have you also looked at that with LDL? Or has that been looked at previously?
Yeah, so that's a great question. And we have. And it turns out that LDL is kind of a protein wasteland. So what we find on LDL is what are called exchangeable apolipoproteins. These are classic proteins that like to bind to lipid, and they shuttle back and forth between LDL, VLDL, and HDL. So we find that subset of proteins in LDL, VLDL, and HDL. But HDL has a whole other set of proteins that do not reside on LDL or VLDL at all, as far as we can tell. And it's even more interesting than that. If we use a technique based on antibodies to isolate the HDL, now we find hundreds of proteins in the HDL complexes. And so we actually think that there's a whole family of very specific macromolecular complexes that are being assembled by protein-protein interactions, not by just binding to lipid in kind of a non-specific way. And these proteins live in this family of proteins that we call HDL. We're even finding HDL particles that are very rich in ApoA1 and have no lipid in them. So this idea that HDL is a protein-lipid complex that floats at a certain density in a centrifuge, we think is an incredibly naive and oversimplified way of thinking about the problem.
You mentioned the antibody study. Have you looked at using micro antibody arrays or something?
Yeah,
in that
so
regard?
these are all things that we're gearing up to do, and one of the things that we'd really like to do is to make these assays more high throughput so that we could start doing clinical studies. So I mentioned earlier that we've looked at people with and without diabetes and with and without obesity and with and without insulin resistance, and we're finding very striking results there. In fact, in this setting, it looks like there's a protein called clusterin, or interestingly, ApoJ, that's very enriched in healthy people and goes down in any of these disorders. And that study involved about 100 individuals. What we'd like to be able to do is to study 300 to 400 people in an analysis, because that's when the clinicians are going to start to pay attention. They don't like these studies that only have 10 or 20 people in them. But our bias is if we don't see any big differences in a small study, it's not worth looking at hundreds of people. We want to find things that are really changing in a major way. And so far, it's looking very promising from that perspective. In fact, Clem Furlow, who's sitting right there, has a major interest in some of these other HDL particles and has developed a lot of these techniques to try and do this in relatively high throughput. So we're actually talking about that right now and trying to come up with better ways to do the analyses.
Great, thank you.
We have other questions? Glenn.
See, if you want a question, just cite somebody specifically in the audience. This is a good way to do it.
Just a couple interesting comments on recent papers. One by Joe Zabner's group in Iowa, where they moved the human paraoxynase gene into Drosophila and showed increased resistance to Pseudomonads, presumably by the quorum sensing factor hydrolysis ability of PON1. So that plugs it again into the innate immunity like the
PON1 is carried on HDL, so that's right. Yeah.
So it plugs it into the innate immunity again like the trypanosome lytic factor.
And Drosophila is a fly model that we use a lot for model system studies. So the fact that you can put a protein into a fly and it kills bugs implies pretty strongly that it's a broad biological function. So these are the kinds of things that we're thinking about.
The other set of observations comes from Jordi Camp's lab in Spain. I don't know if you saw their paper, where they looked at the immunolocalization of PONs 1, 2, and 3 in the mouse, and they found distributions of ApoA1 around the mouse system where the A1 didn't co-localize with PON. And so one interpretation would be that HDL sort of serves as a delivery truck to deliver PON1, for example, and perhaps other proteins around the system. Right. It's quite like carrying lipid, but carrying proteins instead.
I can only say that we completely agree. So we've used antibodies to pull out certain other proteins. One that we're interested in is called phospholipid transfer protein. So this protein, again, is generally believed to play a role in transferring phospholipids between lipoproteins, but its proposed crystal structure just looks like a major neutrophil protein called BPI, bacterial permeability inducing factor, which is a major antimicrobial protein in neutrophils. So it looks like a protein that plays a role in host defense mechanisms. When we isolate these particles using antibodies, and this is work that was done in collaboration with Marion Chung and John Albers, we find almost no lipid in the particles and a whole host of proteins that are involved in complement activation and coagulation. The stoichiometry is very interesting. So there's about, again, clusterin comes into the story. So there's about 8 moles of clusterin, 1 mole of PLTP, 1 mole of ApoA1, and then 1 to 2 moles of a wide array of other proteins that are in these complexes. And when we used a database system that predicts interactions known on documented interactions in the literature, we could show that there were documented protein-protein interactions between every single protein in these complexes. So we're very enamored of this idea that protein-protein interactions are really what's responsible for driving HDL. One of the little problems with all this is ApoA1 may not be the whole story. And so there are HDL particles that are known not to have ApoA1 in them, for example, ApoE. And so it gets to be a complicated story. But we think that it's important to deal with the complexity rather than to scramble them into a random mixture of particles and then call that HDL.
A lot of the discussion, both the medical and lay press discussion of heart disease and lipids, is focused on what are called risk factors and things that you or I do that might modify our risk of developing heart disease or stroke. And I guess there are 2 related questions here. One question is, are there common genetic differences between people in this room, for instance, that strongly influence either the nature of HDL particles or their activity? And conversely, how do the things that we think of as risk factors and that there is some evidence for influences our risk of cardiovascular disease alter these multifunctional HDL particles?
So those are great questions. We really know very little about that. We do know that ApoA1 is an incredibly potent risk factor. So there are humans that lack ApoA1. So they have HDL. They have a normal HDL cholesterol if you do the standard blood test. But they have no ApoA1 in their HDL particles. And there's only been 7 or 8 of these families described, but all of the males in the families develop heart disease in their 20s. And we can recapitulate this in mice by knocking out ApoA1. So we can make mice get extensive atherosclerosis with completely normal levels of HDL cholesterol. So there's very strong evidence from animal models that ApoA1 is important. But it's not that simple. I think this issue of the torcept that increased HDL cholesterol but may be bad for you shows that it's not just a matter of measuring HDL ApoA1 cholesterol ratios. We think it's more complicated than that. We're just beginning to try and dissect out what some of these interactions may be, but really virtually nothing is known. There have been a number of genome-wide association studies that have looked for other things that regulate HDL cholesterol levels, and really CETP comes up over and over again. This is a protein that transfers cholesterol esters between particles, but it has a very modest effect, and it doesn't obviously have a major impact on cardiovascular risk. So I think we're just beginning to try and understand what's going on there. But again, a very interesting idea would be, for example, to do a large study looking at SNPs for SNPs that are associated with cardiovascular disease, and to look in parallel in large numbers of individuals at the HDL proteome and to try and make connections there between influences, genetic influences on the HDL protein composition and cardiovascular risk. To do those kinds of studies, we're going to need high throughput assays where we can do a lot of analyses, because you have to have a lot of statistical power to do that. So I think those are great questions, and we just don't know. We have been throwing HDLs from mice onto macrophages, and we can control the inflammation in mice experimentally. And what we're finding is that what we call normal HDL or control HDL and inflammatory HDL have very, very different effects on macrophages. And this does not involve cholesterol metabolism. So we haven't figured out yet what it is. It could be the proteins, it could be the lipids, it could be both. But at least in our hands, in mice, inflammatory HDL and HDL have very different effects on inflammation in macrophages, and I think that would be another very interesting area to pursue. Yeah.
Thanks again, Jay.